So hopefully you've started to work on your Bio 225 lab report that involves the Kirby Bauer antibiotic susceptibility test. One part of the lab report is to incorporate the procedure. So with this, you need to write out the procedure in enough detail that if a complete stranger read your procedure, they would be able to replicate the test. So you did complete, hopefully, the virtual Kirby Bauer lab, and that shows you kind of how to measure the zones of inhibition and look at the standardized chart to interpret whether the bacterium is susceptible, resistant, or intermediate to the antibiotic. But I wanted to take the time to go over the procedure from the beginning to the end so that you can use this video to help formulate your own procedure for your lab report. So you do not have to have a list of supplies for your lab report, but nonetheless, these are the things that are required, at least in our lab, to do a Kirby Bauer test. So you do need safety gear. That typically includes things like gloves and then protective eyeglasses. You need a Sharpie to label your Petri dish. Whatever bacterial species it is that you are testing, you were emailed with your specific species, whether it's Staphylococcus saprophyticus or Micrococcus luteus, or again, whatever species you were notified that you were required to work with. Various antibiotics. So for our Kirby Bauer test, we're using TMP5, which is trimethoprim, C30, which is chloramphenicol, CIP5, which is ciprofloxacin, and then P10, which is penicillin. These kind of shortened versions are the codes that are stamped on the antibiotic discs, since the names are actually quite long and we can't fit the entire name on those little tiny samples. You do need Mueller-Hinton agar. That's the type of agar in the Petri dish that we use to conduct this test. Sterile cotton swabs, antibiotic dispensers and tweezers, an incinerator, an incubator, parafilm that is kind of like fancy saran wrap that just seals the lid of the Petri dish to the base, a ruler, and then a standardized zones of inhibition chart. So the first part of the procedure is just basic lab safety. So you would have needed to wash your hands, pull back long hair if you do have long hair, put on safety glasses, wear protective clothing, and then obviously wear gloves. So just basic lab safety, you should incorporate that into your lab report. Probably the most important things being wearing gloves, washing hands, and putting on safety glasses. So the first thing that you would do is you would label your Petri dish. So when we work with Petri dishes, and in this specific test requires a Mueller-Hinton agar Petri dish, you would label the bottom of the plate. So we do always label the bottom of the plate, and we would label it with things like the species name of the bacteria that we're using, your name, the date, and often the section that you are in, like Bio 225 CO1 or CO2. But nonetheless, you would need to label your Mueller-Hinton agar Petri dish with a Sharpie. You don't need to go into the details of exactly what you would label it. You just need to mention that it does require labeling. So once you've labeled your Mueller-Hinton agar Petri dish, you're ready to transfer a sample of your bacterial species to that Mueller-Hinton agar. So if you were in lab, I would have given you your species of bacteria growing in a Petri dish. So this is actually a sample of a bacteria known as Serratia marcescens. It often grows this pink color. So if that was your bacteria, you would have been given this Petri dish full of bacteria, and you would have needed to collect a sample from this and transfer it over to a sterile Mueller-Hinton agar to conduct your Kirby Bauer test. So to sample it, we would have just used a regular sterile cotton swab. And when you put your cotton swab into the Petri dish to collect a sample of bacteria, you always want to hold the lid of the Petri dish like a shield. You hold the lid of a petri dish like a shield, kind of hovering over the base of the petri dish to prevent airborne contamination from getting in. So you would kind of angle that lid, you would stick your sterile cotton swab in there and collect a small sample of bacteria. It doesn't need to be a huge scoop of bacteria or anything like that. Honestly, you'd be sufficient just taking one of these colonies with that sterile cotton swab. Then you put the lid of the petri dish back down, and you would go over to the Mueller-Hinton agar, and same thing, you would hold the Mueller-Hinton agar dish lid like a shield while you go in and inoculate and put that sample of bacteria into the sterile Mueller-Hinton agar. Now, when you're inoculating the Mueller-Hinton agar, you want to use what we call a bacterial lawn technique. Basically, that means you would take your sterile cotton swab and go all over this petri dish. So you go back and forth several times at different angles to create that bacterial lawn. Ideally, you want to make sure that you cover every square inch of that Mueller-Hinton agar with bacteria. You want to make sure that there's no part of the agar that has been left untouched, and that will allow you to properly kind of grow a bacterial lawn. Then obviously you would close the lid completely, and you would take your cotton swab that's now contaminated with bacteria, and then you would toss that in the biohazard trash. This sample of bacteria that you were using to collect for the Kirby Bauer test will most likely get tossed in the biohazard trash as well, as you would have been done using it at that point. So now we have a Mueller-Hinton agar plate that has a ton of bacteria present on it because we've inoculated it to form that bacterial lawn. So the next thing we need to do after we formed our bacterial lawn is we need to place the antibiotic discs on the plate. So you would have been given 4 of these antibiotic dispensers. They're kind of like fancy little candy dispensers, but basically, or what are they called? Pez dispensers is kind of what I was looking for. So you would have 4 of these. One would have trimethoprim tabs in it, the other would have ciprofloxacin tabs, the other chloramphenicol, and the last would have penicillin tabs. So what you would do is we typically take tweezers, and we take those tweezers and we sterilize them. We sterilize them in the incinerator that we have mentioned previously before. Once we've sterilized our tweezers, you're going to remove a little antibiotic disc from the antibiotic dispenser, and then you're going to again take your Mueller-Hinton agar dish that you've already inoculated with that bacterial lawn, hold the lid as a shield, and take that antibiotic disc and just tap it to the agar. So now you would have a little antibiotic disc of trimethoprim tapped onto the agar. And they do have the little codes stamped on them, so you'll know what antibiotic you're placing in which spot. Then you would sterilize your tweezers to get any of those extra bacteria off that you may have accidentally got on there, and you would take another antibiotic dispenser that had a different antibiotic. You would collect that antibiotic, maybe penicillin, again go back to your Mueller-Hinton agar, hold the lid as a shield, and then tap your little sample of penicillin on the plate as well. And then you would repeat that with the other 2 antibiotics. When you place the antibiotics on the Mueller-Hinton agar that has now been inoculated with the bacterial lawn, you want to make sure you place them about 4 to 5 centimeters apart from each other and apart from the edge. The thing is, we don't want those zones of inhibition after this incubates to merge into one another. So ideally, each of those antibiotic discs should be placed about 4 to 5 centimeters apart from each other and apart from the edge. So after you've placed all 4 antibiotics on the plate, kind of in that square distribution, then you would need to parafilm the lid of the Mueller-Hinton agar to the base of the petri dish. We would parafilm it, and then again that's kind of like fancy saran wrap, but we parafilm it mainly so that nothing gets in to contaminate our culture. Then you would put it in the incubator. Remember, the incubator is that temperature controlled chamber that encourages bacteria to grow. So we would place it in the incubator for anywhere from 24 to 48 hours. It would need to be in the incubator so that the bacteria have the chance to grow. Hopefully the antibiotics inhibit them from growing, but that's sometimes not the case because sometimes the bacteria can be resistant to the antibiotic. But nonetheless, the Mueller-Hinton agar does need to be parafilmed and placed in the incubator for 24 to 48 hours to allow for growth to occur. After 24 to 48 hours, you would remove the Kirby Bauer plate from the incubator, and you would see the zones of inhibition. Now, you have been sent the measurements for the zones of inhibition for your lab report, so you don't need to physically measure the zones of inhibition, but still, just so you know how this is done, because this will come up on a quiz and on a practical, what you would do is, again, you would measure the entire clear area all the way across. And then you would look at the chart that I have here, and then I also have it in your week 5 PowerPoints to see where that measurement falls as far as susceptible, resistant, or intermediate. But you always measure the zone of inhibition all the way across, and also it should be measured in millimeters. But then again, once you're done measuring in millimeters, you do have to look at the standardized chart that you've been provided with to see if that falls in the susceptible, intermediate, or resistant category. Once you're done measuring the zones of inhibition, you would simply just take your Kirby Bauer plate and toss it in the biohazard so that it could be autoclaved and sterilized. And one more thing, just because as I was watching this I realized I forgot to mention, yes, the plates are incubated for 24 to 48 hours, but one additional thing I do want you to have in your procedure is the incubation temperature. The incubation temperature that we would use would be 37 degrees Celsius. So as you watch this video, make sure that you are, writing your procedure based on what this video entails, and then make sure to ask me if you have any questions about the procedure of a Kirby Bauer test.